names, some of the work which he'll talk about today. So he's a very much a computational biologist with an amazing knowledge of bioinformatics as well. He's teaching me things, which is pretty cool. So he's here today and he'll be talking about systems biology of neglected gastrointestinal pathogens. Thanks, Aaron.
Thank you, Melanie. Before I begin, I'd like to say what a pleasure it is to present this research here at WEHA today, and especially during the centenary for this fantastic institution. And Melanie pointed out to me last night that I actually speak in the middle of the very month of the centenary, and then she added quite friendly, no pressure. So thanks, Melanie. So I have quite a wide range of interests, and basically you could summarize that into an interest in neglected gastrointestinal pathogens, and primarily of humans, although I'm in the veterinary faculty. And this ranges from diarrheal protists, things like Giardia and Cryptosporidium, to gastrointestinal helminths, hookworms, roundworms, these sorts of things. The research I'm going to present today is a lot of the work that we're doing, particularly in soil-transmitted helminths, and in particular in relation to genomics and transcriptomics research, and that's what I'd like to talk about. So to put this in perspective first, when we talk about helminths generally, helminths are, of course, very much an amalgam group. They include tapeworms, they include flukes, blood flukes like schistosomes, they include nematodes. These things are not really related at all from an evolutionary perspective in these broad groups, but nonetheless we call them helminths and we stick them together. And the numbers associated with these parasites are tremendous. 250 million people are affected with schistosomes. We've got 50 million people with taeniid cestodes that Marshall Lighthowers in our faculty does fantastic work on. The area that I'm interested in are the soil-transmitted helminths, where roughly, it depends on who you ask, but roughly somewhere between 1 in 4, 1 in 5 people on the planet are infected with some species of gastrointestinal soil-transmitted helminth. Ascaris, hookworms, whipworms, that sort of thing. When we look at the species, we have a very high prevalence of Ascaris lumbricoides, somewhere in the neighborhood of 1 billion people worldwide infected with these parasites. 90% prevalence in many areas of the world, particularly nasty in young children. Hookworms are a major cause of anemia in children and physical and cognitive stunting as a consequence of that, and whipworms are also very prevalent. And I put a question mark down here. Strongyloides stercoralis is an interesting parasite. It's listed at being 100 million, but the diagnostic tools that are typically used to diagnose gastrointestinal infections with helminths, they look for eggs. And strongyloides produces larvae. And so these techniques don't pick this up. So I suspect this 100 million figure is actually a massive underestimation. Nonetheless, very prevalent parasites. Neglected and often, and historically often ignored because the effects on the host are not, these don't cause a huge amount of deaths. You've got roughly 1.5 to 2 billion people infected, about 150,000 deaths associated with these parasites per year, which given the numbers is not it's still a big number, but given the number of infected is not a massive figure. But what their big effect is chronic disease. They cause massive problems with malnutrition, especially in children, and this leads to long-term physical and cognitive stunting. So if we consider that, firstly, if we look at the distribution, these are tropical parasites, largely through equatorial regions. You've got some differences with the species. Ascarids tend to be more prevalent in Southeast Asia, hookworms more prevalent in Central Africa, but nonetheless distributed broadly across tropical regions of the world, and not coincidentally, obviously, where our largest concentrations of impoverished populations are found. If we look at burden of disease, now this is where the impacts, the chronic impacts of these parasites are so telling. So if we look at the graphic to the far side of the slide here, we see that the overall disability-adjusted life years associated with hookworms, Trichuris, and Ascaris amounts to somewhere in the neighborhood of about 40 million DALYs, which, to put into perspective in some of the research that gets done here, is roughly comparable to the impact of malaria. These are major parasites. Now, if we think about the role that bioinformatics, biotechnology, genomics research can play in trying to understand soil-transmitted helminths and neglected parasites in general, and in trying to find ways to control them as a cause of disease, this paper, which was published in 2002, is quite a nice sort of reference point to illustrate the different ways these technologies can help with controlling disease. And what this was a panel of public health experts that were basically tasked with evaluating the different ways in which biotechnology could contribute to controlling neglected infectious diseases in developing countries. And they basically ranked a whole bunch of different potential benefits based on their overall impact, their appropriateness, their cost effectiveness, their ability to reduce disease burden, the feasibility of their application, all these other different things. And of those, they identified improved diagnostics for infectious diseases as the #1 biggest benefit that biotechnology could provide. And then number 5 and number 7 on the list were directly to do with genome sequencing, being able to do genome-guided drug discovery, and being able to use bioinformatics to generate new antimicrobials, basically. And the point that I've highlighted with these things is that genetics research, genomics research contributes to all 3 of these criteria. Obviously, to be able to develop molecular diagnostic tools, you need to have sequence information. We've done a bit of work in this space in recent years, and this is some of the major publications we've produced. We published the Ascaris genome in 2011 in Nature. The Schistosoma hematobium, this is one of the major African schistosomiasis species. This one is quite interesting. One of the few parasites in the world that's a known carcinogen. It causes urogenital cancers. And it affects approximately 150 million people worldwide. And then in December of last year, we published the whipworm genome, Trichuris. And I'm going to present the research we've done, briefly talk about Ascaris suum. That paper's getting a little old now, but I'll touch on some of the findings we had in that, and then I'll focus a bit more time on the Trichuris genome. So if we look at Ascaris, so Ascaris is the giant intestinal roundworm. Relatively simple life cycle, but with some interesting quirks, which I'll talk about in terms of the genomics data that we found. So you've got an egg that's shed in the feces of an infected host. That gets ingested. A larval stage hatches out of that egg in the small intestine. It moves to the large intestine. It penetrates the intestinal wall, gets into the bloodstream, moves to the liver, then to the lung, is coughed up, swallowed, and then you have an adult stage of the infection in the intestinal tract. Very large parasites. Where's my pointer? Here's my pointer. This bolus of worms was taken from this little girl. You can see this enormously distended abdomen. This is a very high-intensity infection. And something to keep in mind with these gastrointestinal helminths is that their intensity of infection is really heavily correlated with the extent to which they cause disease. A couple of worms, probably never going to notice. A healthy individual from a developed country with good nutrition, a couple of worms, they would never even know. But a small child who's already under nutritional stress because she's living in a highly impoverished situation with a huge amount of worms like this, that's going to have a physical impact just on the function of the gut, blocking of the gut. And one of the major ways that ascariasis can cause severe disease is just simply by causing necrosis of the gastrointestinal tract by blocking the gut itself. They can also climb into the pancreatic ducts, biliary ducts. If you have a fever, they can climb out of your mouth and nose. So they're nasty parasites. Now, in terms of what we did in sequencing the genomes of these guys, I won't go through the details of the bioinformatics. I'm more than happy to talk about this. If people are interested, by all means, please come and see me or ask me after the seminar. But for the purpose of time, I'm going to just focus on the biology aspects. But nonetheless, we basically took a standard approach. We collected high molecular weight genomic DNA from a female worm. We sequenced that on the HiSeq platform. We sequenced a number of different insert size libraries, from short inserts to very large made insert libraries to allow us to sort of assemble through repetitive sequence regions. We used a standard assembly approach, and then we sequenced the transcriptomes from a variety of key stages. And this was we have some excellent colleagues in Ghent University in Belgium that were able to take, this was done in pigs. This is Ascaris suum, which affects pigs, but it's very closely related to the human parasite, and actually will infect humans. And basically what they were able to do was infect pigs and isolate migrating larvae from the liver stage and migrating larvae from the lung stage and then in the small intestine, as well as males and females. So we were able to look at some really fantastic stages of the parasite life cycle in the host itself. And we could then look at the context host-parasite interactions and how they're behaving. And then we did basic standard annotation approaches. We compared to model organisms. Of course, for a nematode, the really important model organism for us is Caenorhabditis elegans, but these other species are also often very useful. We compared them to a variety of standard specialist databases that allowed us to look at things like proteases and kinases and various key protein families that might be of interest to us. In terms of the genome's not massive by a human standard, but large for a nematode, 270 megabases. We were able to assemble this. The main metrics I wanted to show you here is we've got a good, this N50 is basically says that at least half of the assembly is in scaffolds of 408 kilobases or larger, and 90% of the assembly is in scaffolds of 80 kilobases or larger. And basically what you want from that is you want a reasonable level of contiguity in your assembly so that you know that you've mostly assembled complete genes, so when you try and annotate them, you have that information. What we were really interested in primarily is the coding genes, of which there are 18,500. One interesting aspect of the Ascaris genome, our assembly had very low repeat content, 4% repeat content. By comparison, Caenorhabditis is about 20 to 30% repeat. Ascaris undergoes quite an interesting biological phenomenon called chromatin diminution. In the somatic genome, it basically kicks out a lot of repetitive sequence that's present in the germline, but not in the somatic line. We had the somatic assembly, so ours assembled quite nicely and easily, but it's probably about 10% smaller than it ought to be based on this chromatin diminution aspect. There's some fantastic work by Richard Davis's group at University of Colorado where they've actually looked at this diminution and how it changes with different stages of development, but we didn't cover that in our work. In terms of the genome, 18,500 genes, a lot of overlap with Caenorhabditis elegans, which was nice, and various other species and nematodes for which whole genomes are available. And then, as always with these projects, and I'll come back to this later on in the talk, a large number of genes for which we could find no homologue in another organism. And this has some relevance when it comes to looking at drug targeting research, for example. These were distributed across a broad collection of protein families, and we highlighted some that we were interested in. Peptidases have known to play a variety of important roles in gastrointestinal nematodes in their interaction with the host, digesting tissues through tissue migration, also interacting with the host immune system, and obviously involved in digestion in general and feeding. So these are kind of different groups that we were interested in. But the main thing I wanted to show with this was we had a broad distribution of families, suggesting we had recovered the vast bulk of the gene set of this species. And in comparison with Caenorhabditis elegans, it suggested again that we had a fairly complete genome. We then looked at transcriptional behavior in this parasite. And as I said, we were able to harvest larvae during what's referred to as hepatopulmonary migration, that is, in the liver and in the lung, and identified a variety. And this is quite an interesting stage of the life cycle of this parasite as well. You've got a parasite which spends the bulk of its adulthood in the gastrointestinal tract, but at a very short period of its life cycle, but at a very vulnerable period of its life cycle, it's right in the milieu of the body. It's in with the bloodstream, it's in the tissues. It's encountering the full force of the host immune response, and it needs to be able to handle that. It also needs to be able to rapidly get from the gastrointestinal tract through the liver into the lung, because otherwise the immune system will kill it. So we saw a variety of things. We saw a wide variety of secreted peptidases that are known to be involved in tissue degradation in parasites. We inferred that that was involved in helping it move through the tissues. That made sense. We saw, this was, and I'll come back to this in another slide. We saw a lot of chemosensory molecules that are, or homologues or orthologs of chemosensory molecules that are known to be important in olfactory chemosensation, in particular in Caenorhabditis elegans. And we saw a complex mix, and again, I'll come back to this a bit later, of molecules that in a variety of parasitic nematodes are known to play an important role in interacting with the host immune system, in suppressing certain aspects of the host immune response. We also looked at the adult stages. We looked at the male and the female. And because these are such large worms, one of the great things, I mean, adult Ascaris are sort of 30 centimeters at their maximum size, which means you can do a lot of tissue-specific transcriptomic research, which is really nice. And so we were able to actually dissect out reproductive tissue and muscle tissue and compare that side by side. We could look at the differences between male and female worms, but we could also look at the difference specifically in the transcription of genes in the reproductive tract of males, compare that to the muscle of males, identify male-specific genes that we inferred were related to the gonad tissue itself, and then compare that to the female and look at some really interesting differences in the expression of these sex-specific genes. And this was quite nice because most of what we know about these genes is based on research in Caenorhabditis elegans. And Caenorhabditis elegans is primarily a hermaphrodite. I say primarily because there are mutants that are basically male, but more or less it's a hermaphroditic species. So here we actually had something that was dioecious we could look at. And we saw the typical things you would expect to see. We saw genes in the male that were upregulated that have homologs in Caenorhabditis elegans that are associated with sperm and spermatogenesis. We saw genes in the female involved with egg laying, oogenesis, volval development, those sorts of things. When it came to this story, this was something that I thought was really interesting. This is from the larval stages of the parasites, and as I said, we saw this upregulation of a variety of genes that have orthologs in Caenorhabditis elegans that are thought to be involved in chemosensation. And this is kind of neat because the literature, or the modern interpretation of the literature will tell you that hepatopulmonary migration in Ascaris is a passive process. It says they penetrate through the small intestinal lining, they get into the hepatic portal system, they're swept to the liver, they just follow the path of least resistance in the circulatory system. They go to the liver, they get swept to the lung, and out they come. That wasn't a very satisfying answer. This is such an essential part of the life cycle of the parasite. The notion that it's left that entirely to chance. It has the molecules for chemosensation. Why wouldn't it use them? And so we looked at transcription, and what we showed was that there were actually clear, this heat map, everything that's the lighter the color, the higher the expression relative to the rows across, and we saw statistically significant upregulation of different cohorts of genes associated with chemosensation. In the early stages, in the larval stage, a lot of these olfactory GPCRs, chemosensory GPCRs and kinases and things like that are known to be associated with larval development in Caenorhabditis elegans. That made sense. But when we started hitting the liver stage, we saw upregulation of genes specifically associated with volatile chemotaxis, the detection of volatile compounds. And what we know from skin-penetrating nematodes is, because people have done work on how skin-penetrating nematodes interact with the host, and that's known to be mediated by olfactory chemosensation, is that this is primarily in response to things like short-chain fatty acids, ketones, that sort of thing. One of the key molecules is something called urocanic acid. And one of the things I found quite interesting about that was that the organ of the body, aside from the skin, that produces the most urocanic acid is the liver. So there's these chemosensory cues appear to be sort of present, and we see an upregulation of a variety of chemosensory genes that are associated with volatile chemotaxis, and we think that that's playing a role. Obviously, we have to now test that, but that's what we think is happening. And then we see, as we hit the lung stage, upregulation of things like NPR1 and TAX2, and these are known to be in C. elegans associated with aerotaxis, the positive response to oxygen. And that, of course, makes a lot of sense. If you're heading to the lung, that seems a pretty logical cue to pick up on. So it seems to us that this is, in fact, not a passive process, that this is, in fact, an active process. The parasite is able to sense where it is in the body, and it uses that to help it guide its way through. And there's some anecdotal evidence to support that. One of the primary experiments in this field that's been done is where in pigs, you established experimental infections in pigs where the researchers injected larval Ascaris underneath the skin into lymph nodes, into atypical locations they wouldn't normally go to in a standard infection. And yet they still went to the liver and the lungs. So it's not just as simple as they go through the small intestine, they get into the hepatic portal system, and they're swept along. And in fact, the other side of that is that the larval stages actually go into the large intestine, so they're not immediately coming into contact with the hepatic portal system anyways. Now, if we move from the Ascaris genome, I'll spend a little bit more time on the Trichuris genome, and this was published in Nature Genetics in 2014, so just this past December. And Trichuris is an interesting parasite. So we have two sides to this parasite. We have human trichuriasis, which is quite a nasty gastrointestinal disease. You have a very simple life cycle. You've got an egg that's shed in the feces, that gets ingested, then it develops in the small, firstly, this larval stage embeds into the epithelial cells of the small intestine, then it moves into the large intestine, and it sits in the large intestine. And it does this kind of neat thing. It's got this long, very slender, modified esophagus called a stichosome, and it basically pushes that underneath the superficial layers of the epithelium, and it feeds. In terms of disease, again, malnutrition, a competition to the host for resources, but some of the main things, hemorrhagic bloody diarrhea, and I'll touch on this. No one really understands why these things cause bloody dysentery. One of the nastiest symptoms that I think you could associate with a parasitic disease, you get a tremendous dysentery, and ultimately something called rectal prolapse, where the sphincter muscles collapse and the rectum telescopes out of the body, which I think is the most unforgettable symptom of a parasite. I spared you and didn't put a photo on there, but trust me, it's nasty. So we basically sequenced the genome of this. We looked at the male and the female, and again, we also looked at transcription of different stages. We were able to get larval stages that were embedded in the small intestinal wall. We were able to get adults, and we could actually separate the adults so we could look at the expression of genes in the stichosome relative to the body. And there was two reasons we were interested in this. One of this was obviously to understand this parasite as a cause of human disease, and the other was this other interesting aspect with Trichuris suis, the pig parasite, which is this notion that there's this relationship between the human immune system, the way the immune system develops, and exposure to parasites, and in particular to helminths. This idea that if you're not exposed to these parasites, this is the so-called hygiene hypothesis that I'm sure you're familiar with. If you're not exposed to parasites at an early age, your immune system doesn't develop in quite the right fashion, it becomes dysregulated, and you get this emergence of a wide variety of autoimmune disorders. And Trichuris suis ova has actually been used and is currently being used as and explored as a biological treatment for autoimmune disorders, along with hookworms. Alex Lucas's group at James Cook University does fantastic work with hookworms. This is another example of this. And this is a company in the United States called Coronado, which is trialing these eggs. You ingest the eggs, you get the larval stage of the parasite. It doesn't develop to adulthood, you don't get any disease, but it's able to suppress a lot of the inflammatory responses associated with a wide variety of gastrointestinal disorders. One of the latest, Coronado did a very large phase 2 trial on, I think it was Crohn's disease, which didn't seem to be very successful. And so there's debate as to exactly how this works. And I think ultimately, you know, it's a matter of identifying the mechanism that this parasite is tweaking rather than a concept of a biological control where you give someone an egg and a parasite. Most people parasitologists don't seem to have a problem with that, but everyone else does. Nonetheless, so we were interested in both aspects. We were interested in how does this thing cause disease, but also how is it interacting with the host immune response? What's it doing? So again, same story. We got DNA. We did sequencing on the HiSeq. We used pretty much the same approach of what was done for the Ascaris genome. And we did the same sort of annotations. And then we also sequenced the transcriptomes from eggs, from larvae in various stages of development in the gut, from the adults. And we looked at alternative splicing, and then we also, this was a bit of a different ad, we also did small RNA, small non-coding RNA sequencing on these as well, and found some interesting things. I'll talk about that. And then we were able to sort of compare the relationship between particularly microRNAs and expression profiles. So in terms of the genomes, these are much smaller than the Ascaris genome. They're sort of in the neighborhood of 80 megabases, quite compact. Our assembly overall was about 80 to 75 megabases, which is sort of standard with any sort of next-gen short-read sequencing project. You usually seem to get about 10% of the genome that you can't assemble. And then we had about 75 megabases collect into large, nice scaffolds. Large N50, large N90, fairly complete sequence. Much higher repeat content, but that didn't seem to have any impact on our assembly, which was nice. In terms of the genes, the male genome had about 14,500 genes, the female genome about 1,000 left. I got really excited when I saw this because there's actually a different XY karyotype. Most nematodes are XX for females and XO for males. That is, they have an X chromosome and nothing else, and that's how they make males and females. Trichuris is the standard XXXY. So I thought, okay, this is great. We'll have 1,000 genes associated with the X or with the Y chromosome. We'll be able to identify that. No one's really looked at that in nematodes too much. But that wasn't the case. It turns out that the gene content, the actual gene content between these doesn't differ, and what differs is simply the copy number of some genes. And we infer from that that the Y chromosome of Trichuris is mostly repeat content. One of the major things we were interested in, given this interaction between the hosts, that you've got this very intimate interaction where the larval stage embeds itself into the tissues of the intestine, or you've got a sticosome that's embedded underneath the epithelial cells and engaged in digestion, we were quite interested in what sort of proteins does this parasite produce that it secretes from the body. So we looked at typical secreted proteins, and that basically is is defined as the way we did it anyways, was defined as a gene that contains a signal peptide domain for export across a membrane, doesn't contain a transmembrane domain, and in our case, was present, or a homologue of that gene was present in proteomic data that's been generated for a variety of helminths from their secreted products. That's how we kind of defined that. There are atypical secreted proteins that don't have a signal peptide domain. Those are pretty difficult to predict bioinformatically. We put those to one side. Based on that, we identified 618 excretory secreted products that we were pretty confident were genuinely secreted products. A lot of them are proteases, particularly this family, S1, chymotrypsin-like or trypsin-like proteases, and I'll talk about them in the next slide because they're quite interesting. A wide variety of secreted transporters, ions, small molecules, things that like sugar and nucleotide capture, transporters, that sort of thing. These porin proteins, porins have been shown, these are barrel-forming toxin proteins. They've been shown in Trichuris trichiura, which is the human parasite, this TT47 protein is from that they actually generate pores or holes in the host cell membrane. And so it's thought that they kind of give access to the parasite to the cytoplasm of their host epithelial cells. And we found a wide variety of these, quite a rich family. These pinins, I'm not sure about these ones, but I highlighted them because they were kind of interesting. There were 9 of these things in the secretome, and they had really high homology with humans. And why I kind of thought those were interesting is that these pinins are involved in cell-cell adhesion. And I thought, well, perhaps there's some sort of docking thing happening here where the parasite protein is mimicking host proteins to try and fix itself on or get a more intimate contact with the host epithelial cells. Don't have any proof of that. But nonetheless, there's this high degree of homology. And then a wide variety of immunomodulators, and I'll talk about those. So when we look at immunomodulatory proteins, and I'll come back to this again later in the talk, what we saw was quite interesting. With Ascaris, you have quite a broad range of proteins that are involved in a wide variety of functions. They're trying to stimulate alternative activation, or at least we infer this based on their homology with proteins where this has been demonstrated in other species. They have a wide variety of proteins that are involved in stimulating alternative activation of macrophages, in inhibiting neutrophils, in inhibiting antigen presentation by dendritic cells, in stimulating promotion of FOX regulatory T cells, a whole wide variety of things. Trichuris was a bit different. With Trichuris, it was much more tightly defined. You have a few molecules, in particular TPX, thioredoxin peroxidase, and macrophage inhibitory factor, which stimulates alternative activation of macrophages, which in other helminths has been shown to block macrophages from producing nitrous oxide, and instead promote them to produce anti-inflammatory IL-4 and IL-10 cytokines, which downregulates the toll-like receptor pathway. Toll-like receptor pathway is known to be dysregulated in a lot of autoimmune disorders, so that was quite interesting. We also see some molecules such as a TGF-beta, what appears to be a vertebrate TGF-beta mimic, and a cysteine protease inhibitor that are known to block antigen presentation on B cells and stimulate the promotion of pro-inflammatory T cells. We see calreticulins and apyrases, which mop up extracellular ATP and calcium. And again, in other helminth species have been homologs of these have been experimentally demonstrated to help to block the inflammatory response. And lastly, we see this one, this serpin, which is quite interesting. This was actually from TSCEI, this cathepsin elastase inhibitor, and TS is Trichuris. So this has been experimentally demonstrated in this species as being an important molecule in blocking these major proteases that are associated with tissue remodeling and lead to both tissue destruction and the promotion of an inflammatory response as a consequence of neutrophils. And this is interesting because I'll show that neutrophils actually play an important role in the immune response against this parasite. And all of this, just in case you're interested, this is all inferred based on what's known in helminth immunoregulation in other species, and this is a fantastic review from James Hewitson and Rick Maisels that was published in 2004, I think that was. No, that was 2009, sorry, in Mall Biochem Parasitology. The other group that I mentioned that was really interesting were these S1 trypsin-like serine proteases. So this is a predicted structural model of one of these proteins. And it's got your standard chymotrypsin or trypsin barrel protein domains. And we saw these things being produced in abundance in the secretome. And when we looked at and I'll touch on the transcription of these things, we saw that they were highly transcribed. And we were very curious about what they were actually doing. And there's some interesting aspects to this which kind of point to maybe some potentially interesting mechanisms in these parasites. One was this experimental study, not done by us, but done by Hasnain and PLOS NTD, where they showed that Trichuris, when you infect rodents with Trichuris, they produce something, they didn't define what it was, but they worked out that it was a protease, that degrades the mucus barrier. It's a really important component of the host defense system, one of the first barriers against pathogens in the gastrointestinal system. And specifically, these things were digesting mucin 2 polymers. And they showed that they could block this when they used trypsin inhibitors. So they inferred this was a trypsin-like molecule, but they never identified what it was. So it made a lot of sense that we were seeing trypsin-like secreted proteases as being one of the major components of the secretome. And this is potentially really fascinating because one of the things that's been shown in humans and in other animals is that there is some sort of genetic, you have people that get high-intensity infections and people that get low-intensity infections. All parasites in general are this underdistributed model where you've got some individuals get a lot of parasites and then a lot of individuals get very few parasites. There actually appears to be some genetic link to this in helminths, where you can actually show that individuals who get treated for nematodes and then reinfected will either establish a high or low infection. It correlates with whether they had a high or low infection previously. And it looks like part of the sort of genetic component to this is actually the ratio of mucin 2 and mucin 5 polymers that you have in your mucus barrier. So these things seem to attack mucin 2, but if you have more mucin 5, it's not able to degrade that mucus barrier so well. We also showed that in general, trypsins are involved in breaking down protein polymers, and one of the other polymers that seemed one of the closest homologs to this structure in humans was kallikrein, which is important in taking prekallikrein and making bradykinin. And bradykinin is an important molecule in stimulating the inflammatory response. And I have no proof of this, but I wondered if maybe part of the role of this parasite was degrading this message and making something other than bradykinin and trying to block that inflammatory cascade. I don't know that that's the case. When we looked at transcription of these molecules, we showed that in the larval stages, when they were embedded in the tissues, we saw an upregulation of a lot of these secreted proteases and immunomodulators. One of the things we saw that I found quite interesting was an upregulation of lactoceramide synthesis. These are sphingolipids. And I didn't know what beta-glucosyl-ceramide was, but when I had a look, I was quite surprised to see that sphingolipids have been explored heavily as anti-inflammatories and are used as treatments for a wide variety of gastrointestinal autoimmune disorders. And this is some cellular work that was done in a paper where they stimulated in a mouse basically an IBD response, an inflammatory response, and they showed that when they used beta-glucosyl-ceramide, not from Trichuris, but just beta-glucosyl-ceramide generally, they could actually reduce that inflammation substantially. So they seem to have some important anti-inflammatory molecules. We also looked at the sticosome, and we were interested in what the genes were being produced by the sticosome. We argued that if there were genes involved in regulating the host parasite inflammatory response, they ought to be upregulated in the sticosome tissue relative to the body, which is just sitting out in the intestinal lumen. And we saw that that was the case. We saw a wide variety of those. And we saw these porins and various other things that sort of fit nicely with what we were expecting to see. So we got quite a lot of insight into the biology of these guys. I want to move through this quickly because there's a couple of other things I'd like to talk about beyond the genomes. But we also looked at non-coding RNAs in this species, as I mentioned. We sequenced basically from the same species or from the same stages that we sequenced our transcriptomic data from. We sequenced all non-coding RNAs of sort of 50 nucleotides to 25 to 50 nucleotides in length. We identified a variety of small interfering RNAs, or at least we think we do, based on their mapping patterns. We found ones that appear to be involved. These Pee-wee RNAs or something like them have been described in Caenorhabditis elegans as being important in blocking transposable elements. We saw other RNAs that don't have this 21 nucleotides. A 21U RNA is something that's 21 nucleotides long and it starts with the uracil residue. We didn't see those, but we saw other ones of a similar length that started with a G that we think might be doing the same thing. They tend to be found antisense to transposable elements, and they seem to be upregulated, particularly in the male germline. And they may play a role in suppressing, in protecting germline integrity. That's been shown for peewees for 21U RNAs in Caenorhabditis. And then we identified 320 microRNAs. And basically what we did is I defined a unique RNA based on a unique mapping location, because you do get quite a lot of variation within these reads. They map to a single spot, but there might be nucleotide morphology. And no one really has discussed polymorphisms, to my knowledge, in small RNA. So I basically said if it maps to one spot, that's one RNA. If it maps to one nucleotide over, that's another. And again, I can talk about the bioinformatics of this later. We looked at relative expression of these microRNAs, their abundance, and we showed that there were different clusters, and particularly high abundance of microRNA transcription in early larval stages, and then some consistency. This is the adult male, and this is the male posterior body, and this is the adult female and the female posterior body. This is the part of the body that sticks out in the lumen, the vast bulk of the body of the organism, and you see consistency in that. And then you've got a few microRNAs that appear to be specific to or at least highly upregulated in the sticosome. And a lot of these were ones based on homology with Caenorhabditis elegans. Let7 is a really important regulatory microRNA in a wide variety of nematodes, so those were to be expected. These were kind of things that we expected to see. We also tried to look at correlating, because with microRNAs, you should see generally an inverse relationship between microRNA transcription and the mRNA that it's supposedly regulating, right? MicroRNA goes up, the mRNA gets suppressed, it drops down. So firstly, we predicted microRNA binding sites, and then we filtered that because predicting microRNA binding sites is really don't want to say sloppy, but I can't think of a better term right now. There's a very high level of false positives with this. Probably 90% of the things you detect are not actually real. And so we wanted to try and filter that. And so what we did was we said, okay, well, we've got this fantastic model in Caenorhabditis. So what we'll do is if we have a microRNA that is transcribed in Caenorhabditis, it's got a homolog in Caenorhabditis, and we're predicting an interaction with a homolog of a coding gene in Caenorhabditis elegans, and we have evidence from research in Caenorhabditis elegans where they've actually looked at genuine microRNA, mRNA binding interactions that support that prediction. Then we'll take those and we'll look at those, and everything else we'll filter out as being too likely to be a false positive. And when we did that, we saw some really cool stuff. When we looked at in particular, when we looked at the male and female, we saw a downregulation of about 320 genes that coincided with the upregulation of this MRI, or the microRNA 228. And what was interesting was that these genes that were downregulated in the males were largely associated with feminizing functions, vulva development, germline, egg production, embryogenesis, things like that. In the female, we saw the opposite, upregulation of these two mRNAs, or microRNAs, and downregulation of a variety of genes enriched for things like spermatogenesis, male mating development, etc. This was kind of neat, because this suggested to me that perhaps the natural state for the nematode germline is hermaphroditic, that all genes are being transcribed, and then there's a layer of microRNA control that goes on top of that that suppresses the opposite gender phenotype, which was kind of neat. The last thing we showed with small RNAs, one of the things you do with small RNAs when you're looking at them is you look for these kind of patterns, things that are overabundant, things that don't quite fit. And there was one clearly glaring pattern here. These were these small RNAs, non-coding RNAs of 21 nucleotides. They started with an A, and they were incredibly abundant in our data set based on their transcription relative to their actual abundance in terms of unique sequences. Wanted to know what they were. So we looked at where they were. They were distributed generally throughout the genome, roughly half in coding and half in non-coding regions, but when we looked by transcriptional abundance, 85% were transcribed in non-coding regions, and they were almost always on one strand, which was quite interesting. I don't know how that works, but nonetheless, that's what we found. So we said, well, what were these things? We had a look, and they had some different structural motifs associated with them, and they had transcriptional patterns, particularly a high abundance of transcription in the male germline. We don't know what these things do, but we think that they're a novel class, might be a novel class of nematode-specific non-coding RNAs. No idea what their function is. And there's evidence that these exist in other nematodes. We see these in other data sets for small RNAs, but people haven't described them before. So I'm going to switch gears now. I'm going to talk generally, the last bit of the talk, on, pardon me, move back into one of the original purposes we discussed in terms of why we do this research, which is to try and understand or try and develop methods that improve control of gastrointestinal helminths. And this study that talked about some of the different ways that this sort of biotechnology data can contribute to this effort to control these diseases. And as I said, one of the big things was drug target discovery. So there are two basic approaches in which people use genomic data to identify what's called the druggable genome, which is genes that are thought to be potential targets for inhibitors that could be used for parasite control. One of those methods is essentiality prediction, where what you do is you identify homologs in model organisms. You go and look at the knockout phenotypes for those genes if they're available for those model organisms, and you triangulate among a number of different model organism species and say, okay, well, if that gene is present in my case, for Ascaris, for example, in Caenorhabditis, Drosophila, and yeast, and it's a lethal phenotype, if you knock it out in those three organisms, it results in the death of the organism, then odds are pretty good that if we were able to knock it out in the parasite, which is very difficult to do in most of these parasites, it would probably be lethal as well. The other approach is this metabolic choke point analysis, where what you do instead is you look at the metabolism of the organism, you look for enzymes which fulfill a unique function. They uniquely take a metabolite and convert it into something else, or they produce a unique metabolite that they're the only source for. And you say, okay, well, if I attack that, then I'm attacking a unique function, and either it's going to lead to an overabundance of some metabolite or the starvation of some downstream pathway. In the case of the Ascaris genome, we tried both and combined them together. I predicted essential genes by cross-comparing lethal phenotypes with Caenorhabditis, with Drosophila, and with yeast. And then I filtered those and said, okay, now how many of those are associated with a metabolic pathway that I can find no redundant function for based on metabolic metabolic pathway analysis. And we identified a whole variety of these things, and some of them were quite interesting. I then filtered this for single copy genes and said, okay, well, it'd be interesting to work on those because they're going to have less allelic variability, and we kind of narrowed this down to 5. And now I'll tell you why I don't trust any of this data. One of the basic way that you use this is, not that I don't trust it, but just that there's some caution to be had with this. You identify, there's different ways you could sort this, and there's probably a better order to do this in, which is to actually identify a compound that kills and then use bioinformatics to try and infer the target. Whereas I think target-driven, I think most people in this room will probably agree that target-driven drug discovery, which is identifying a target and then trying to find that one inhibitor that's going to specifically kill it, is pretty unlikely to be successful, or at least it's going to be very difficult to do. Nonetheless, you identify an essential target, you try and identify known inhibitors, you try and predict structural interaction some way, and then you go and test, or you flip this around and start with testing first. One of the problems with this is you're trying to find a motif in the protein where your inhibitor sticks to. And the challenge with this is I was interested to know, well, what's the relationship between an essential gene and alternative splicing of genes? Do essential genes tend to be very rarely spliced, or do they tend to undergo a lot of exon shifting and changing? And I thought it would be that they would be very rarely spliced because they're essential. If you change them, you're going to disrupt their function. You can't do that. It turns out it's the opposite. So when we look at nematodes in general, Caenorhabditis produces about 1.5 transcripts per gene. It has 20,000 genes. It produces about 30,000 spliced transcripts. Parasitic nematodes seem to have expanded that significantly. You get about 2 transcripts per gene on average in Ascaris, and in Trichuris, you get about 3 transcripts per gene. This is inferred based on RNA-seq data, but nonetheless, that's what it shows. And when I looked at that in Trichuris, and I did the same essentiality prediction, I found that on a 3-to-1 ratio, essential genes were multi-isoform, they produced multi-isoforms. They were alternatively spliced relative to non-essential genes. And that was quite interesting because that sort of suggested to me that you need to know, if you're going to use this drug target approach, you need to know if you're going to target a particular motif, how conserved that motif is. When that transcript isoform is being transcribed, is it being transcribed, not just the gene, but the isoform that you're targeting, is it being transcribed in the stage or in the context that you want it to be? That's quite important, I think. We also looked at this chokepoint, and I have comments on this. So, as I said, chokepoint analysis is this idea of identifying these bottlenecks, these unique genes that have a non-redundant function. The problem with that is that in any parasite genome project, you wind up with these huge numbers of genes that have no homologue in anything, and even a greater number of genes that even have a homologue in another organism but have no known function. We don't know what they do, right? And so then, generally speaking, you've got 20 to 50% of parasite genes that have no annotated function, no known homologue at all, or if they have a homologue, it doesn't have a functional annotation associated with it. About half of the genome is metabolic function. It's not very difficult to extrapolate. Maybe not half of these unannotated genes have a metabolic function because we know metabolism fairly well, but nonetheless, it seems reasonable to expect that there's a goodly portion of these hypothetical proteins that have some sort of metabolic role. We don't know what that is. So when we start saying these are bottlenecks, there's a lot about the metabolism that we don't know for these. There are different ways that we can improve on this, and these are some of the different examples. There's Metapsych and Biopsych, which are better databases for curated metabolic pathways that provide more information than, say, for example, the KEGG pathways, which people use. You can do cluster analysis where you can align genes based on, so you get fragments of genes, you align them based on sequence similarity, and you transfer annotations from one to another to try and flesh out. You can do pathway analysis where you look for co-regulation of genes or co-transcribed genes and cluster them and enrich them under the hope that your genes will be functionally enriched if they tend to be transcribed in similar contexts across the database. Or one of the things that we've been playing with a lot lately is inferring homology based on structural homology of the protein rather than sequence homology. And a fantastic program for this is something called iTassr. And iTassr is a program that uses a wide variety of structural prediction software. You give it a protein sequence, it predicts conserved domains, it folds it, and then it tries to align and wire that structure to crystallized structures available in the protein data bank. And this is quite a nice program to use. There's a benchmarking sort of organization or conference, if you will, for inferring structures of proteins called CASP. And iTassr is perennially at the top of the list of being the most accurate program for defining protein structures. And then you can map this up. And this is an example that one of my students, Brendan Ansell, who did his honors here at WeHi, did for Giardia, one of the parasites we're interested in. This was a protein that had no known function. It had no structural or no sequence homologue that we could identify. He took it and he put it through ITASR and made the blue structure is the model, and the white is the protein that we folded, and it matched beautifully with cytochrome P450 reductase. So well, in fact, that you could even add the cofactors onto the structural model. Now, this is an in silico experiment, but I'd be willing to bet pretty heavily that this is cytochrome P450. This is computationally very intensive. But we've been doing some work with Victorian Life Sciences Computing Initiative that allows us to actually do this on a genome scale. In general terms, it takes about, you can do this online. There's an ITASR server. You can take your protein of interest and go there, and you can annotate your protein, but you do them one at a time. It takes about 3, depending on the size of the protein, 3 to 4 days to annotate. I want to do this for thousands of proteins, and so that obviously is something I can't do on a server. But I've been able to get access to a substantial amount of computational resources through VLSCI, and I'm now doing this for Plasmodium falciparum and Plasmodium vivax. And that's underway. Last thing I want to talk about is the other side of the story with gastrointestinal helminths. As I said, there's this interaction between the parasite and the host in terms of the immune response. And this is being explored as a possible both relationship with and therapy for autoimmune disorders. So as I said, we have this story. Gastrointestinal nematodes somehow influence the balance of the immune response from a Th1 to Th2, promoting a Th2 response at the sacrifice of Th1 and Th17. And this is kind of projected as the story for gastrointestinal nematodes. And the point I wanted to make really quickly was that to ask this question, well, are all helminths the same? Nematodes are an incredibly phylogenetically diverse phylum. And parasitism has arisen within the nematoda on at least 5 to 7 different evolutionary time points, right? Independent parasitism arising from free-living nematodes through to arthropods, and then finally into animal and plant, particularly animal parasitism. How then can we say that these things are all the same? When we look at their biology, we look at Ascaris. They have different life cycles. Ascaris undergoes hepatopulmonary migration. Trichuris does not. Hookworms do. There are different components. I won't touch on this too much because we don't have time, but there are different components to the way that they interact with the host depending on where they are and whether the host has been encountered them before. You've got an IgA and mast cell response in early intestinal infections with Ascaris. Later on, you get eosinophilia in the lungs, for example. That changes whether they've had prior exposure or not, and so on. When you look at hookworms, again, you've got hepatopulmonary migration, but you've also got the fact that at the adult stage, they're not just sitting in the intestine feeding on, bits of food and some bacterial fauna. They're biting down in the intestinal wall and sucking blood. Their interaction with the host is different. And you do see differences in terms of the cytokines that they stimulate, the different antibody profiles, things like that, when you see a hookworm infection compared to an Ascaris infection. And same story with Trichuris, where again, now it spends its entire life cycle in the gastrointestinal tract. So if we look at the immunomodulators, if we look at Ascaris, as I said, Ascaris has an incredibly complex sort of strategy for dealing with the immune response of the host, where these are all the different molecules that have been known to be associated in helminths in some helminth species or another with some way of influencing the immune response, suppressing, as I said, stimulating alternative activation of macrophages, suppressing neutrophil responses, suppressing antigen presentation on dendritic cells, and so on. And when we looked at the immunomodulators, the homologs of these proteins that Ascaris produced, we saw that they produced pretty much all of them, which kind of makes sense because it basically goes everywhere in the body that you could pretty much go and encounter the immune system. You're in the bloodstream, you're in organs, you're in the intestinal tract. Same story with Necator, but Necator does something that's different. So strongyl nematodes produce this enormous family of something called SCP taps, and these are also referred to as venom allergen-like proteins, and they come up a lot in a wide variety of snake venoms, for example, and they're known to be very allergenic molecules. Ascarids don't produce these, or very few of them, and Trichuris doesn't either, but strongylids produce hundreds of them. These must influence in some way, shape, or form that interaction with the host. We look at Trichuris, and we have, as I said, a much more pared back immune response system where you've got basically a focus on a couple of key pathways that are really heavily directed towards the inflammatory response. So it seems safe to say that you can't just simply say the story about TH2 is promoted, TH1 and TH17 is suppressed is an oversimplification, I think. Yes, that happens, but there is very much a species-specific difference, and not understanding that without genomics makes that very difficult to sort of reconcile. So if we look, basically this is just saying what I've already said. We've got different antibody profiles. We've got a different focus, a complex immune reaction when it comes to Ascaris and hookworm, not so much for Trichuris. And this becomes very important to understand. Firstly, if you wanted to develop vaccines against these parasites, you need to be able to, this is my second to last slide, you need to be able to understand how they're interacting with the host. You need to know whether they develop acquired immunity, how do they interact with the host, what are they suppressing, what's the major component of the immune response you want to stimulate. The other side of it, which is quite interesting as well, is you also have to understand that these parasites are ubiquitous, and they're playing a role, and a species-specific role, I'd argue, in suppressing aspects of the immune response. And then if you want to come into these areas that have high prevalence of these parasites and develop a vaccine against another pathogen, these things have an impact. And these are some papers, these are quotes from a couple of recent papers on developing, in this case, vaccines against HIV and tuberculosis, in the bottom, vaccines against Plasmodium. And the first quote says, "Helminths are strong immunomodulators, and negatively impact on the ability of the infected host to mount a protective vaccine-specific T-cell immune response for HIV and other pathogens." And the bottom one says that the future of malaria vaccine development programs need to account for worm-mediated hyporesponsiveness to immune reactions. We need to understand the biology of these organisms at a species-specific level. Genomics provides us with an aspect to allow us to do that if we're going to be able to get a handle on how these ubiquitous parasites affect these other parasites as well. So I'll close with that. I'd like to thank all of you for your attention. I'd like to acknowledge a huge team of people that have helped me with this research, some absolutely excellent scientists, and of course, fantastic funding and other types of support from a wide variety of institutions. Thank you very much for your attention.
Any questions for Aaron?
You think if we're able to develop vaccines to these helminths that it might also help with autoimmune diseases, or is that safe?
I don't know how that would work. What I can say is that where we've seen, traditionally the way that, and at the moment, the way that these parasites are controlled is through chemotherapeutic treatment. And where you see a wide-scale use of anti-helminthic drugs and a drop in helminth prevalence, you actually see now an increase in a wide variety of autoimmune disorders. There's actually studies that have shown that if you take kids that are infected with helminth and you treat them with, say, benzimidazoles, which are one of the most commonly used drugs, mebendazole, for treating gastrointestinal nematodes, they actually become more reactive to a wide variety of common allergens. So probably I would guess that if you developed a vaccine for helminths and you purged helminths from the environment, you would see an increase, not a decrease, in autoimmune disorders, but I don't know.
So you've identified a number of genes you think regulate the immune response. Do you see their transcriptional profiles differ as the organisms, but for those that can move between different environments, alter?
Absolutely you do. Yep, definitely. So in the case of Trichuris, for example, we saw that the highest transcription of these what we think are immunomodulatory proteins occurred in the larval stages when they were embedded in the epithelial cells, and in the adults were found in the sticosome, which is in the tissue rather than the body. And one of the things we saw that was interesting, I mentioned this cathepsin elastase inhibitor that blocks cathepsins and elastases in neutrophils. And in our data, that peaked in the larval stages that we harvested 4 weeks after the experimental infection in pigs. And that's an interesting time point because in the histopathology work that's been done on the immune response against these parasites, the neutrophil response peaks at 4 weeks. So they coincide beautifully. So yes, absolutely. And in Ascaris, same thing. The migratory stages, we saw much higher transcription of these genes than in the stages that were in the intestine.
Aaron, I was wondering, give us a state of play on what's happening with some of these potential immunomodulatory molecules and then being used as therapeutics. How far along are these things, and what's the state of play?
So there's a couple things, and this isn't my research, but nonetheless I'm aware of it. TSO, the Trichuris suis ova, Coronado produces that. There's a company in Belgium that you can purchase these things from. They're in various stages of clinical trials. The latest clinical trial I know for TSO was the work that was done in, I believe it was Crohn's disease. It was either Crohn's disease or ulcerative colitis, and that wasn't successful. Separate to that, Alex Lucas at James Cook University has done a huge amount of work on this with hookworms, both in infecting individuals with hookworms themselves. And he's got some beautiful data on celiac disease, for example, where they show after 6 months of a hookworm infection, a celiac can eat an entire bowl of pasta without any problem. It's tremendous. In addition to that, I know he's identified a couple of key proteins that he thinks are really important, and they're working on that. And I believe they're working on that with Johnson & Johnson, but I don't know the details of where that's at. But definitely that's moving. And I think that's the way to go. I think infecting an individual I think one of the problems they had, and I suspect one of the unknown quantities, is with a biological organism, the TSO that they're using, these things have been produced in culture for years and years and years. Okay, well, in an infected animal. But nonetheless, how do we know that they've been stable in whatever it is they do, which we don't know? And how do we know that by giving an egg to someone that that's going to produce the same response every time? Whereas if you give them a protein or something like that, you know the concentration, et cetera. I think you've got a more reproducible model.
All right, I think we better wind it up. I'd like to thank Aaron again for a fantastic seminar.